All right, today we are going on an absolutely incredible journey. We're not just diving into the human brain, we're actually going back in time, billions of years, to uncover the ancient and honestly pretty surprising story of a single, powerful molecule, serotonin. And this story, you'll see, starts in a place you would never, ever expect.
So, let me ask you this. What on earth could your mood possibly have in common with 2.5 billion-year-old bacteria? I mean, it sounds like a joke, right? How could your complex feelings possibly connect to some of the earliest life on this planet? Well, the answer to that question is really the key to understanding why our brains work the way they do.
And to find that answer, we have to rewind the clock. I mean, way back. We have to start at the very, very beginning with the origin of the system itself. Just look at this timeline. It really gives you a sense of the massive scale we're talking about here. The story of serotonin, it doesn't start with humans, it doesn't start with animals, it doesn't even start with brains. Nope. It starts 2.5 billion years ago, with ancient bacteria just floating around in the primordial oceans.
So picture this. These ancient little organisms, cyanobacteria, they developed this protein called rhodopsin, and its job was pretty simple. Just capture sunlight for energy. But the key part of this little light-catching machine, the part that actually did the work, was an amino acid you've probably heard of, tryptophan. Now I want you to file that name away, tryptophan. It's going to be really important later. And here it is, our first huge clue. The modern serotonin receptors that are in your brain right now, they are the direct evolutionary descendants of those ancient light receptors. Scientists call this substantial homology, which is just a fancy way of saying they still share a big chunk of their original design. It's like an unbroken biological chain stretching back billions of years. It's wild. Okay, so let's jump forward a bit. How did this system go from just detecting light to actually sending signals between the very first cells of the very first animals? I want to introduce you to the Hydra. This little creature is one of the earliest animals to even have a nervous system, and it's a perfect, simple model for us. Even back then, serotonin was already a key player, acting as one of the very first neurotransmitters. And the system it used, it was beautifully simple. A sensor cell makes and releases some serotonin. That serotonin then just kind of drifts over to its neighbors, and then specific receptors on those neighboring cells pick up the signal, and poof, you get a response. That simple three-step dance, release, travel, detect, that is the fundamental blueprint for literally all neural communication. It all started here. You know, there's this classic saying in biology, ontogeny recapitulates phylogeny. It's a mouthful, I know. But all it really means is that the way an individual develops from an embryo often mirrors the evolutionary journey of its entire species. And we see that perfectly here. Just like these serotonin receptors showed up super early in the story of life, they also show up incredibly early in our own development as embryos. In fact, some are at their highest levels before we're even born. But here's the thing. This early success, it created a brand new problem. A really big one. A central challenge that would basically dictate the next billion years of brain evolution. Hey, remember tryptophan, that key ingredient from our ancient bacterial ancestor? Well, here's the catch. For animals, for us, we can't make it ourselves. It's what's called an essential amino acid, which means we have to get it from our food. And get this, it is the only thing the body can use to make serotonin. No tryptophan, no serotonin, period. So this is the crucial point. The raw material for this absolutely vital signaling molecule has always been hard to come by. It is, from an evolutionary perspective, very, very expensive. And that scarcity forced an incredible evolutionary trade-off. Think about it. On one hand, the chemical signal itself, the serotonin, is rare and precious. So what do? You can't waste a single drop. Evolution's answer was brilliant. If you can't make the signal stronger, you make the antennas better. You evolved an incredibly diverse and super sensitive set of receivers to make sure every last molecule gets heard. And right here, this is the big reveal of our whole story. The reason you have so many different types of serotonin receptors in your brain isn't some random accident. It's a direct, elegant, evolutionary solution to the ancient problem of tryptophan scarcity. The whole system is designed for maximum efficiency with a very limited resource. So this whole strategy of building better and better receivers, well, it also drove huge changes in the delivery system itself, the actual physical wiring of the brain. Now, the source material for this explainer lays out this fascinating evolution of the serotonin network's physical layout. It tracks the changes all the way from a simple sea slug to a complex primate. Let's break down the key stages of this amazing transformation. So first up, you've got what the researchers call the autocratic or bull approach. Think of simple creatures like the sea slug. They have just a few very large neurons that make these huge, sprawling, widespread connections. It's like a fire hose, powerful but not very precise. It's basically a broadcast signal to a huge area. Then as you move to animals like rodents, you see what's called a committee organization. Here, the neurons start to group together into bigger clusters, and their wiring, their axons, bundle up to travel to the front of the brain. It's definitely more organized, but it's still a pretty broad approach, a kind of general invasion of signal. And finally, we get to the primate brain and what's called collaborative organization. Now, this is where things get really sophisticated. The neurons are in smaller, more specialized groups, and crucially, their axons become myelinated. That's the fatty insulation that lets signals travel way, way faster. This system isn't a fire hose anymore. It's built for precise, rapid, selective strikes. So the whole evolutionary trend is crystal clear, isn't it? The whole system, driven by this constant pressure to make every single molecule of scarce tryptophan count, it evolved from a slow, blunt instrument into a fast, precise surgical tool. Which brings this whole billion-year story right back to where we started, right inside our own heads, and to the incredibly complex legacy that we've all inherited. So after all that, after this billion-year saga of evolution, what are we left with? What is the final result of all this pressure to diversify and get more efficient? Well, it's a number. This number, actually. Over 20. That's right, over 20. That's how many different types of serotonin receptors are active in the human brain. And every single one of them is a chapter in this epic story. Each one is a finely tuned instrument evolved over 1 billion years to respond to a precious, scarce signal. It's an unbelievably complex and nuanced system, and it's all thanks to tryptophan being so hard to find. So I guess the final takeaway is this. The hardware that runs our moods, our anxieties, our social connections, it wasn't designed overnight. It was forged over billions of years by sunlight, by scarcity, and by the raw drive for survival. So when you realize that our emotional toolkit is literally that ancient, well, how does that change the way we think about our own minds today?